Hello everyone, I am Dr. Sirajul Faisi. I welcome you in my YouTube channel. Today in this video, I am going to talk about colorimetry and spectrophotometer. You know, my friend, many biological experiments involve the measurements of compound or group of compounds present in a complex mixture. Also, the most widely used method for determining the concentration of biochemical compound is colorimeter, which makes use of the property that when white light passes through a colored solution, some wavelength are absorbed more than other. So many compounds are not themselves colored but can be made by absorbed light in visible region by reaction with suitable reagents. So these reactions are fairly specific and in most cases very sensitive, so that quantities of materials in the region of millimolar per liter concentration can be measured. The big advantage of is that complete isolation of compound is not necessary, and the constituents of a complex mixture such as food, blood can be determined after little treatment. So the depth of the color is proportional to concentration of the compound being measured, while the amount of light is proportional to the intensity of the color and hence the concentration. So the earliest colorimeter relied on human eye to match the color of a solution with that of one of the series of color discs. The results obtained were too subjective and not particularly accurate. So colorimeter is generally any tools that characterize color sample to provide an objective measure of color characteristics. In chemistry, the colorimeter is an apparatus that allows the absorbance of a solution at a particular frequency, color of visible light to be determined. Colorimeter hence make it possible to determine the concentration of a known solute since it is proportional to the absorbance. Now, friends, different chemical substances absorb varying frequencies of visible spectrum. Colorimeter rely on the principle that the absorbance of a substance is proportional to its concentration. That is, a more concentrated solution gives a higher absorbance reading. So, filter is the colorimeter is used to select the color of light which the solute absorbs the most in order to maximize the accuracy of the experiments. Note that the color of the absorbed light is the opposite of the color of the specimen. So, a blue filter would be appropriate for an orange substance. Sensors measure the amount of light which has passed through the solution compared to the amount of entering, and display read the amount absorbed. Now friends, the quantitative reading for the concentration of a substance can be found by making up a series of solution of known concentration of chemical under study and plotting a graph of absorbance against concentration. By reading off the absorbance of the specimen substance on the graphs, the value for its concentration is formed. So, white light from a tungsten lamp passes through a slit, then a condenser lens to give a parallel beam which falls on the solution under investigations contained in an absorption cell or cuvette. The cell is made up of glass with the sides facing the beam and cut parallel to each other. So beyond the absorption cell is the filter, which is selected to allow maximum transmittance of the color absorbed. If a blue solution is under examination, then red is absorbed and red filter is selected. Note the color of the filter is complementary to the solutions. Now friend, how colorimeter work? The light then falls on to a photocell which generate an electrical current in direct proportional to the intensity of light falling on it. Means a small electrical signal is increased by the amplifier which passes to a galvanometer of digital readout to give absorbance reading directly. So among the simplest and most common colorimeter are Spectronic 20 and Spectronic 21, commonly called Spec 2 and Spec 12. Now friend, spectrophotometers is a sophisticated type of colorimeter where monochromatic light is provided by prism. The bandwidth of light passes by a filter is quite broad, so that it may be difficult to distinguish between two components of closely related absorptions. With a colorimeter, a spectrophotometer is then needed. So, light source of and prism, cuvette or simple holder, photo multiplier, and output meters. See the percentage transmittance, elliptical component. Now, all types require a blank, which is a solution that contains the entire reagents except the substance to be measured. All types require a blank, which is a solution that contains the entire reagent, except the substance to be measured. It is used to adjust the device to 0. So here is a summary of the steps of operations of Spec. Power, turn on power, allow about 5 minutes when first turn on, warm up, then wavelength, select appropriate wavelength, then 0 with simple holder, empty and close, adjust meter needle to percentage transmittance using 0 control knob. Now blank, fill tube half full with water, place in sample holder and close cover. Adjust meter needed to 100% transmittance or 0 absorption using light control knob. So standard, measure absorptions of known solutions. Fill the tube half full with a sample of known concentration. Place in the sample holder and close cover. Read absorbance value from meter. Repeat this step if making a calibration curve or verifying proportionality pairs. So simple, measure absorbance of solution with unknown concentration as in previous step. Now types of spectrophotometer, visible spectrometers and ultraviolet UV spectrophotometer. Now colorimetric determinations of vitamin B12. Vitamin B12. Vitamin B is water soluble vitamin, RDA 2.4 mg per day for adult. It is found only in animal source. Functions: aid folic acid in synthesis of heme. It prevents anemia required for protein digestions and absorptions. Now you can see you have the blank 1, 2, 3, 4, 5 and unknown. AUNK means unknown before blank. Now results, you can have the concentration at x-axis and absorbance at y-axis. So calculations: absorbance of unknown, concentration of unknown, absorbance of standard and concentration of standard. So my friends, I hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.